Hello and welcome to today's NanoServe webinar about the PicoBalance. We will concern ourselves how we can measure mass of single cells and beads, and that is masses that are in the nanogram and picogram scale. And we'll discuss some applications of this method of measurements. My name is Gotthold Fläschner, and I have been part of the research team that has been pioneering this technique in the recent years in the biophysics laboratory at ETH in Zurich, and I'm soon going to join NanoServe to help them bring this technology to the labs around the world. Let's dive in immediately. So first, very general. So why is mass of interest? Why is it important? Well, mass is a base unit to begin with. That is, many units you might know are derived from like these base units. So a set of very fundamental units. And one of these units is mass. And it's being used all over in engineering and natural sciences. And the reason behind is that mass characterizes systems quite well. So you can use mass, for instance, to quantify amounts. You're familiar with that if you like cooking or baking. But it can also be used to measure sizes, for instance. So it can be a very nice proxy for volume, in particular if you have complex shapes. Now, it is not only interesting to characterize, to characterize or quantify, but it is also interesting because it affects the speed of processes, for instance, in chemistry. It also affects structural integrity of certain structures. So it is something that is interesting because it can affect other things and is being affected by many other things. So it's a kind of integrative value or property of a system, as we'll also see later on. Or actually, as we can see also right now, if we think about it in a cell biological sense. So what can you do with mass, in particular if you know the mass of a single cell? Well, what you can study is how a single cell grows. So growth is actually a complex process. It is regulated. The cell needs to know when to stop growing. Otherwise, it's an abnormal growth. You find such things in cancer. And that is a process that is affected by many things. So it might be the environment, really, like what kind of nutritions are around. It might be the history of the cell. It might be the internal of the cell, how it looks like that. So this is something that you can study quite well, in fact, if you know the mass of a single cell, because looking at it microscopically is actually very tricky, or actually rather impossible to do with an accurate measure. So the uncertainty that you have in your volume estimate there is rather big. And if you want to make it small, you're running into trouble, because this you typically can only do with high intensities of light. With this technology, which I'm going to present to you, you can actually measure quite accurately, less than 1% of the cell mass, in fact, how the mass of the cell is changing, and that is how the cell is growing, for instance. And at the same time, you have an environment for the cell, which is like a cell incubator or physiological environment. So the cells just keep on growing. They don't notice that are being measured. Now, you can study like this, the growth regulation, as I pointed out, but what you can also do is for non-growing cells, let's say, how they are in homeostasis. So for instance, you can have a look at cells that as part of the physiological function, they secrete or take up water. These kind of measurements have been done in tissues, but now you can also do that for like on a single cell level. Now, this is, of course, in interest on a fundamental level, like growth regulation, but it is also important, both of them actually, because you also always have the flip side of the coin. So what is if those things do not work properly? Because then often diseases are the consequence of that. So if you want to study diseases on a single cell level, actually knowing how the mass of the cells that are affected by the disease is of great interest. Now, I will discuss these examples in more detail once I've come to the applications here. But at the moment, I want to first tell you about the concept of the measurement and the implementation of it. So let's start with the concept. If you want to measure the mass of yourself, typically what you do is you just step on a scale, on a balance. Now, this is basically measuring how much gravity is pulling you down onto that scale and basically measures the weight force. Now, this concept is quite handy for things that are our size or bigger, but they don't scale that well. So if you want to measure a single cell in a liquid, you can't really do that with weight force. So what you're going to do instead, or we have done so, is to use inertia. In effect, not the gravitational mass, but the inertial mass. What does that mean? Well, mass boils down to affecting the resistance against acceleration. So if you have a heavy object, it is hard to accelerate it. That is hard to make it move fast, let's say, or stop it, in fact. So if you think of, for instance, a car, let's look at this car, let's accelerate it and let's stop it. And then it's accelerating in the other direction. So it's moving back and forth. It performs a certain oscillation movement. Now, if I make that car heavier, well, you already know what to expect. This cannot be done that fast anymore. It's just trickier to accelerate something that is heavy. Now we can take this concept and scale it down, like not this concept, but the car, let's say. So what you would do is, for instance, take a cantilever beam. So that's just like a diving board that you would find somewhere in a swimming or near swimming pool, but just scaled down by a lot. So this thing, like the length would be probably like the width of a hair, that is 100 microns. What you can see is that the cantilever here, the beam, is just performing an oscillation. So if you look at the free end of it, it's just going up and down, up and down. So it's oscillating, it's accelerating and deaccelerating, like just the car that is just driving next to it. So and the idea would be now, like if I attach a mass to that tiny cantilever, then this vibration frequency would be affected by the mass that is being attached. So this is our concept, but we need to formulate it a little more precise, like what is being affected really if we attach a mass there. So qualitatively, it becomes harder to really accelerate the cantilever there if there's higher mass. But what we want to measure is the natural resonance frequency. So the natural resonance frequency is the frequency at which the cantilever would oscillate if there was no damping. So, and we can find out what kind of frequency this is if we apply a force, a periodic force to the cantilever beam and write down how the cantilever beam responds to that. For instance, we can have a look at the amplitude. So we are exciting, that is putting a certain force onto the cantilever with a certain frequency. So we are poking it fast. And we just see what the amplitude is. That is the response amplitude of the cantilever. Now we go to different frequencies. So for instance, we poke it a little faster. We see a different amplitude, a higher amplitude in this case. So we kind of get the idea, it might be easier, it might be nicer. With the same force, we get a higher amplitude of the cantilever. So we're getting towards the direction where the cantilever actually likes to oscillate better. Now, another thing that we can have a look at is not only the amplitude, but the phase. So to every corresponding amplitude that we have just, pointed out here in the plots, we have a phase. Now, the phase basically describes the relation between the excitation and the readout signal. So we excite, this poking thing, the oscillation, we excite with a sine wave, and we read out a sine wave. But the question is, like, are they in sync? Are they synchronous or are they displaced in time? So, and if they are not displaced in time, we would have a phase of 0. And well, then for instance, here we would have a displacement in the two signals, and that would be a phase that is then non-zero. We can measure at many different frequencies and end up with this kind of curves. So this you might have seen somewhere, that would be an amplitude curve. So here would be the amplitude resonance, that is for the same given force, so we don't change the magnitude of the force which we use onto the cantilever, but we change the resonance frequency. This here is the highest amplitude of our response. Now, that is, you might think, also the resonance frequency, the natural resonance frequency, but it's not quite, because as I said, the natural resonance frequency is the one that is there without damping. Here we have damping in the system because we have a liquid around. So what we can do, instead of just reading here the maximum peak, which is not quite right in a liquid, we would just fit these either the amplitude or the phase curve, and would extract the natural resonance frequency. By the way, if you would be in not in a liquid, but for instance in air or even vacuum, then this amplitude resonance, that would coincide with the natural resonance frequency. But there's another way actually to extract the resonance frequency than just by fitting these curves. What you can also do is have a look at the 90-degree of the phase. So there where the phase is 90 degrees, that is that the excitation and the readout signal are just displaced by 90 degrees. There is where you have your natural resonance frequency. And that is, as I said, not quite where the peak is, but just slightly before that. So we're left with two different ways how to get that natural resonance frequency, and that is fitting amplitude and or phase, or get the frequency at which the phase is 90 degrees. That is, that they are just offset by that amount. So the idea, just to generally like recap, is you have a certain cantilever that is vibrating at a certain frequency without any mass attached, and then you attach a mass to it, and it's changing that resonance frequency. And knowing what the resonance frequency before and after the attachment of that mass is, allows us to determine the mass according to this formula which you see here. K is hereby the spring constant, that is the stiffness of the cantilever. 4 pi squared is just a factor. And then you have the resonance frequencies here before and after, like inverse squared and subtracted from each other. Now, there is one more thing. So this simple formula actually already tells you the mass of something that you attach. Not quite though, because there is an additional factor. So this formula is true actually only if you attach the mass at the very top of the cantilever, so at the very free end. If you don't have that, you do need to consider the sensitivity of the cantilever. So you can imagine the sensitivity is the highest. It's actually 1 at the end of the cantilever, and it's quite low somewhere like here, so more close to the base of it. But we'll discuss that later. The very general idea, just know the resonance frequency before and after, and well, then you can calculate your mass. Now, how is that being implemented? So we know now the concept. How do we actually do all what I said? How do we, for instance, poke the cantilever? I just told it like this. You can't really poke the cantilever. Your finger is too big. This cantilever, as I said, is really tiny. So what typically one does is exciting cantilevers with a piezoelectric crystal. Now, that is basically a crystal that is vibrating, moving, if you apply a certain voltage, a changing voltage to that. Yet, so this you would place near the cantilever, and this vibration would propagate, would go to the cantilever and would make it vibrate. Now, the curves that you extract out of that are actually not so good. So you can see here a phenomenon that is called the forest of peaks. In black, you see the amplitude, for instance, and on the left-hand side, and on the right-hand side, you see the phase. And what you see is really something called forest of peaks. That is that that you really have a hard time to determine what the proper curve is, how it looks like. And the reason is that this excitation with a piezo is rather dirty, let's say. So what you can do to get a cleaner excitation is what we did with with a laser. So you have a laser that is focusing at the base of the cantilever and is heating that base periodically. And that periodic heating causes a periodic movement of the cantilever, which can be read out by an additional laser. And now this is now a recording that we have on our instrument, and you see here the data, it fits very well, or the fit fits the data extremely well. So it's really like a textbook excitation. So it's very, very clean compared to this phenomenon of the forest of peaks. And this now allows us to determine, for instance, the phase, 90 degrees phase properly, or to fit the curves well. Now, how does that look like in an experiment? So here you see a model of the DriveAFM. Below here you see a dish filled with a red liquid, and that is the medium in which the cells will be housed. So we have prior detached the cells. So they have been growing in an incubator. They have been growing, for instance, in cell culture flasks. You would add some trypsin, for instance, to make them loose. Then you collect them and then you put them into this small Petri dish that is below this instrument. And now let's see what we do. So here you see in green the cells. Here you see on top the cantilever. So this is the chip that holds the cantilever. Here now we excite it with that laser that is focusing at the base. And we see the amplitude of the vibration, which here is widely exaggerated, but what we need to know is actually just the resonance frequency. Now we measure the resonance frequency again after we have picked up the cell and see how this resonance frequency is changing over time as the cell is, for instance, here growing. Now the cell sticks to the cantilever as we have before coated it with a sticky protein. So cells like to live, for instance, they like to attach to extracellular matrix proteins such as fibronectin or collagen and other like matrigel or whatever you can think of. So and these we put onto the cantilever, such if we have these cells that are rounded from the trypsin before that we've put into the Petri dish, such that if we touch them, they like to stick to the cantilever, and there we are. We can then see how this attached mass is changing the resonance frequency of the cantilever over time. Now, this might look like, for instance, here, if we just record the phase and the amplitude, with a lock-in amplifier to be technical. So you would see here now white dots appearing if we have started the measurement, and you see then the resonance here, the amplitude, and here also in the phase. And you could fit that to extract your resonance frequency. And then if you want to go further and look at how the mass is developing over time, what you can just do is the phase lock loop that is seeing how the phase is changing over time around this 90-degree value. And then you can see how the mass is developing. Now, this is the implementation and the concept. Now, let's go to the applications of it. For instance, we have been interested how cells would grow over time if they are healthy and if we infect them with a virus. So what we did is we picked up with a collagen-coated cantilever a HeLa cell, that is an ovarian cancer cell, and we let it spread, that is flatten on the cantilever for some time. Now, probably this is important to point out here. Everything we do and see here is just on this very instrument. So if we have the cell, if we manipulate the cell, if we pick it up, everything is compatible with light microscopy. So you really do see at the same time what is happening in an optical microscope. And that was also very helpful here, because here you see the cantilever and there is a cell spread on it, which you at the moment can't see due to the contrast and our focus, because what we have focused here is cells that are spreading at the bottom of the dish. And in this case here, we have a cell that is expressing a green fluorescent protein, and that is because it has been infected by a vaccinia virus, which just encoded that protein, like this green fluorescent protein. Now, what we did was just to go in contact with that infected cell to naturally infect the other cell, the healer cell. We would just go in contact, wait a bit, and see how actually the growth behavior of that infected cell and the healthy cell would differ. Now, this is what we saw for a healthy cell. So you would see here in black the growing mass over time. Here you see the spread cell. After 8 hours, it actually rounds up and goes into mitosis. And then afterwards, you have 2 cells. And so it goes on for about 30 hours when we stopped the measurement. So what now happened to a cell that we infected the way that I just described before, that is going down onto the dish and staying in contact with an infected cell? Well, this looked like this. So you would see in the mass curve that the cell would just not grow anymore. It would just stay relatively constant. And at the same time, we would see in the microscopy images that a green signal would come up over time at the same time as the cell now is starting to produce the virus in itself. So now here it starts the interesting bit, like why is that the case? Why does the cell stop growing actually? Because, well, we do see that there is a stop in the cell cycle, so there was no mitosis anymore, but it is actually not clear, or it's actually not always the case, that a stop in the proliferation also results in a stop of growth. So the question, in fact, here would be, is the cell actually prevented, or does the virus stop the growth of the cell and therefore halts the proliferation because there is some kind of growth regulation of the cell that waits to reach a certain size threshold, or what is happening here? So this is something that remains for further investigation. But what we could see here clearly is that there is a significant difference between infected and healthy cells. And this difference we saw really in our mass traces. Now, what we see here as well is now a different example, not the HeLa cells, but the MDCK cells. And here the green is a different thing. This is just the histones being tagged. And the reason for that histone tag is to see better when mitosis is happening. So here you have just seen it, the cell rounds up, and here you see the metaphase plate of it. So, and then the chromosomes, they make 2 parties, so to say, for, I mean, the cytokinesis, they split such that you have 2 cells later on that can spread. And this is just like how the proliferation of these cells work. And you see that over time, it actually comes up, or will come up some more times, because, you know, we're at 16, 17, 18 hours. And this movie goes on for some more time, and you really see that the cells are happily dividing on that while we are measuring them, in fact. And so while this example is proving that we actually have a very good condition for our measurements for the cells, what we also see is that there are dips occasionally coming up in our mass measurement, and that is something to watch out for, because this is not, in this case, the real mass. What I told you before is that that we want to see how the mass of the cell is affecting the resistance against the acceleration of the cantilever, and that actually only works well if you have whatever you're looking at well attached to the cantilever. And now during mitosis, what happens, as you see here, or we'll see in a moment, is that the cell actually is hardly be seen because it's very flat. So you have hard time to see the borders. But now as it rounds up, you can much more clearly see the cell here. And the rounding up at the same time actually relates to a losing of the attachment in these areas. So it's really balling up. And in that moment, what is happening is that we see a reduced mass. So then we'll get an apparent mass here. This is the dip where the mitosis is happening, because we lose the contact, or the cell loses the contact to the cantilever. Yeah, let's proceed, or actually talk a little bit about how we can achieve that the cells are so happy over time that they go through several generations. And to just tell you that we are using a certain chamber that we have built here at mimics an incubator. So you have really the Petri dish, in which you have later on your cells and in which all the measurements will be performed. And there's a silicone lid that is when we're in measurement position with this cantilever holder, it goes down, then this thing is sealed. So you have a sealed chamber, and you have a second chamber around, which you float in like humidified air with a certain CO2 level, such that the pH of the medium is controlled well enough. And you also have a heating inside, such that the cells are, just all cozy. So this is really like an incubator. That this incubator, this mini incubator, enabled our long-term measurements, but we also can do short-term measurements. Now we've done these also with HeLa cells, this time attached differently. So we would have concanavalin A around these, or attached, absorbed to the cantilever, and we would just stick the cell to that concanavalin A. It's just sticking to sugars, in fact, on the outside of the cell. Now, and what we would see here is now, or look at, would be a time frame of 40 seconds and not 40 hours anymore. And you see here a fixed cell that is nothing biologically happening anymore. That is basically a representation, or not a representation, that is the noise level of the machine. So here you see what is going on if there is no biologically active substance on the cantilever. So you would just see that grayish line, and that has a certain width, and that is the noise of the machine. But now what is happening if you really have a living cell? Well, what you see here is that the black with the overlay of red and green curves. So the green and red are just a guide for the eye to tell you, like, there's several things going on. There are fluctuations of the mass within the 40 seconds that you can roughly split into slow and fast fluctuations. And what you can also see is, and that is the actually very interesting bit. And if you wonder yourself, like, what is that? What is happening there at such a short time scale? Well, we found that out by actually depriving the cell of its energy. And we would see when we do that, that these fluctuations, in particular the slow one, is vanishing, and also the fast one is decreasing by a lot. So it is an active process inside the cell that somehow results in that fluctuation in mass. But what we could further on determine is that it has something to do with water transport. So if we blocked aquaporins, so basically the pores through which the water can shuttle through the membrane of the cell, then we would see the same phenotype. And that is really just a flat line that you see is not like if the cell is dead. So there is some more things going on, but is also clearly very different from these fluctuations that we have seen without the blockage of the aquaporins. So here we can actually really make use, or have made use of, the instrument's high resolution, that is the high time resolution, and also the high mass resolution. In time, we really have 10 milliseconds resolution, so there's lots of things you can play with and investigate, and the mass resolution is 5 to 10 picogram, around that ballpark. It can be even put lower, as we will see later on. It very much depends on the cantilevers that you're using, and also the amplitude that you're using for your measurement. So, but nonetheless, if we're talking about a few picogram, then this is significantly less than the total cell mass, which we see here is around 2, in this case, 2 to 3 nanograms. So really rather 1,000 times higher than what our resolution here is. Now, what can be done as well is not just short-term and long-term measurements. We can also do snapshot measurements. That is to look at a distribution of masses. So you just measure different masses, for instance, here of HeLa cells and fibroblasts. And you would see here a fibroblast being attached to a cantilever. And you would see here that, for instance, the population average is just smaller for the fibroblasts, and also that the distribution is slightly different than that of the HeLa cells, indicating that the fibroblasts are just a little smaller than, like on average, than your ordinary HeLa cell. What you could also do is actually talking about small cells to go for way smaller cells. So we've been just seeing like mammalian cells and saw how they behave or how they differ in distribution sizes. But this was always in the ballpark of a few nanograms. Now, yeast cells are interesting because they are way smaller for our measurements, and they're interesting biologically because they are a very well-controlled system. So there are many questions you can answer with yeast cells because they can be just engineered to the degrees that you want to ask or answer certain questions with. So the question is, can we also see yeast cells and their mass on our instrument? And the answer is obviously yes, we can do that as well. In particular, if you use a little smaller cantilever beams. Because with smaller beams, smaller beam size, you get a higher sensitivity, so you can detect smaller masses. And now what you see here is just a budding yeast over time, so you can see in the DIC how a bud is appearing, and here in the mass, so in the DIC I mean differential interference contrast microscopy, so you just look at it with a microscope, and what you see at the same time is here the mass going up over time. And I can't tell you much more on this one here at the moment because this is a manuscript that is under review at the moment. Hopefully it comes out soon. But it basically investigates how cells, or how yeast cells in particular, the budding yeast, is growing in respect also to the cell phase or the cell cycle phase. Now, the examples I've shown you so far have been relying on attaching or gluing, let's say, the cantilever to, or the cell to the cantilever. The glue and sometimes is the same, and sometimes it's the same glue that the cells would use inside the body to glue them to the tissue or inside the tissue. That would be the extracellular matrix proteins. But we can do also differently. We can use a micropipette as used in fluid force microscopy. Here you see a cantilever that has a small aperture at the end. And the cantilever itself is hollow. And now what you can do is you can apply a negative pressure, that is a suction at the end of the cantilever. And now being in liquid, you would be able to just suck, or so in air, you would be able to suck a cell of interest to that aperture, go up, do your measurement, or remove it, or spit it out somewhere else. Now, and this, for instance, we have applied to some nuclei of liver cells. And here you see, for instance, red liver cells. Here you would see such nucleus. And in this case, we would have measured the nucleus to be, or we have measured the nucleus to be also quite small compared to the mammalian cells, like the full cell, weight about 0.24 nanogram. Now, we have also used this cantilever, or you can also use this cantilever to do something quite nice, and that is to have an easy way of a position correction, because this is something I only hinted at in the beginning when I said it is important where to attach the mass of interest. So because, where is it coming? Yeah. So the thing is, if you have a cell, for instance, spreading here, it is important where it is. So if it is in the very end of the cantilever, then you don't have to apply a correction factor for the mass. So this, you might remember, is the formula that I had shown you before, where the resonance frequencies are plugged in and the stiffness of the cantilever that gives you a mass. But the thing is, there is a correction factor if the mass is attached somewhere else than at the very end of it. So, and that is typically the case. So you see here how the sensitivity, or how this function psi here is developing over the length of the cantilever. So you see it is quite high at the end. It's 1 at the end, and it's 0 at the very end, and somewhere in between in the middle. So if you, and that is also kind of intuitive, if you would attach a cell or a mass or bead, for instance, at this part where actually there is no vibration amplitude of the cantilever, then you would not read out a mass, and then your correction couldn't even save you there. But if you attach it somewhere there, then you just have to multiply a factor of, for instance, 1.05 to get the right mass out of there. Now, how to get the position here that you have to plug into this function here? Well, there are several ways to do that. One is to measure optically where the cell and it has its centroid, so really where the center of it is, the projection of the cell. This can be really done with the microscopy. Alternatively, what you can do is with the said beads, with the said micropipettes, is that you actually don't have to know where they are, because you know that there is an aperture at a certain place where basically the mass is being attached to that particular place of the aperture. So really then you know what the distance is. Or, and that is also something that we have been exploring lately, is to use, and that is more for the experts, something that is called a multi-mode excitation. But that basically just says that you sample at very different frequencies, how the amplitudes behave, and how the resonance frequencies behave in very different regimes. And what you can do then is determine the position of the cell or the attached mass, not optically and also not by just putting it somewhere where you know it, but you can measure it non-optically. So you can get out the position of it, so you can plug this into your correction factor and then get the mass regardless. All right, so we've also used that, for instance, to measure beads. So different silicon oxide beads. You see here that we will just, so it's hardly be seen here, because it's not on the focal plane, but it will soon be in the focal plane, the bead. So you have a bead here below that cantilever. So here you see the cantilever, here somewhere is the aperture, and then we start the sucking, and whoops, there it is. There is the bead that is now attached to the cantilever. And now it can be measured in its mass. And what we find out, and that is quite interesting, here you see different bead sizes and therefore different masses, is that actually the measurement uncertainty with a mass is using our device lower than if you rely on a determination that is just optical, so, or that actually the manufacturers would give you. So you can really determine the distribution of these beads quite accurately and with low uncertainty. And yeah, I mean, meaning the mass or the size, if you also know the density of that particular bead. Now, wrapping it up, coming to conclusions. I've told you that mass is one of fundamental importance in engineering and science, because, or natural sciences, because it is really a parameter that integrates many properties. Let's think about growth, for instance. It is affected by the nutrition. It's affected by how healthy the cell is, as you see. So it's really an interesting parameter, therefore. And yeah, it's really of fundamental interest. It can be used, it can be measured if you were talking about like big things via weight force, but we can't do that for tiny things ourselves. So what we look at is the inertial mass. So we try to see how difficult it is to accelerate a beam like a cantilever with a mass attached to that, to really see how hard this acceleration is, which gives us the information about what the mass of the attachment is. Now, the question is, how do we excite these cantilevers? So how do we make them move? How do we make them oscillate? This we do with the clean drive, or which is a photothermal excitation. So you have two lasers in play, one for the readout, that much is a standard thing, but you have an additional laser at the base, and this is introducing the movement via a thermal heating cycle. Now, what I further on told you, that there is a very high time and mass resolution that we can achieve with that instrument. That is 10 milliseconds and just 5 to 10 picograms, even lower for yeast cells if you take smaller cantilevers, or in general if you take smaller cantilevers. And everything that we have seen and done can be done, or has been done, in cell culture conditions. So there is no effect on the health really of the cells, unless you want them to be unhealthy. That is, if you, for instance, infect them with a virus or stress them in any other way that you want to see them stressed, and see how this affects their mass behavior. Now, talking about that, I've shown you some applications, and that were, for instance, like the static measurements, for instance, like they're seeing what the population or how the population of beads look like, what the distribution is, or for fibroblasts and HeLa cells, how this population looks like. We've seen short-term measurements for HeLa cells, that is in the 40-second range. We have seen long-term measurements over 40 hours. We have seen mammalian cells, that is here shown HeLa cells, fibroblasts, MDCKs, but really there is you can do way more than this. It's just if you can find something that you can make it stick to, or actually if you use the fluid FM cantilever, not even that, you can just stick it to the cantilever and measure its mass. So we have also seen not only mammalian cells, we've seen yeast cells, which are way smaller, and even there we were able to get a nice mass readout. We were able to do cell nuclei and beads. We gained insight into a growth. So in this case, that was in the long-term measurements for the HeLa cells. We saw how healthy cells grow and how virally infected cells do not grow anymore. We have seen, although I didn't discuss that too much because, as I said, the manuscript is still in the preparation, how the yeast cell cycle is affecting the cell growth of yeast. You have seen also an example of how you can go into the research of homeostasis, because we have seen in the short-term measurements how water is being shuttled across the cell membrane that caused our fluctuations, which of course needs way more research, but we were able to see that really it was the aquaporin blockage and the reduction of energy content in the cells that stopped that fluctuation in mass from happening. And what we also saw is these size distributions, which are also set. So with the fibroblasts, the beads or nuclei, so you really can get a little bit of an idea about how your population looks like. Yeah, then there are, of course, way more interesting research questions that can be posed with that instrument, really things that live from several things that we, for instance, have a very high resolution, that we're compatible with light microscopy, so you can do other assays at the same time. What you also have is that everything is happening in cell culture conditions. So yeah, if the cells are not affected by your measurement. So really, there's many things you can do with already these few conditions with these things I just mentioned to you. So maybe if you like, we can discuss now some of these applications that you have in mind, or about the instrument, about its functionality. But for now, I thank you very much for your attention. And whoops, I was too far. And yeah, let's open the Q&A session now.